The time in which we live is certainly one of intrigue. The widespread availability of antibiotics and vaccinations has opened the gates of modernity, drastically reducing disease for those with the access to them. Freedom from the shackles of pestilence has allowed for the average human life expectancy to increase by entire decades in the time of what is now one human lifespan. To put it into perspective, before antibiotics, bacterial meningitis, an infection of the spinal cord and brain, killed over 90% of children who contracted it. Now, the mortality rate has been decimated, down to just 5 to 10% of children. This is truly a proud, incredible feat, and seems to be another piece of evidence of humanity's domination over nature. Unfortunately, as usual, nature finds a way to put us back in our place. Due to the principles of natural selection and evolution, the use of antibiotics is becoming less effective the more that they are used. Any bacterium that has randomly mutated resistance to a given antibiotic is able to survive more easily within it, giving it the opportunity to reproduce rapidly, as bacteria tend to do, and pass on its genes, creating a population of highly resistant bacteria that stronger antibiotics must be used on in order to be effective. Eventually, we may not have antibiotics strong enough to combat even simple infections because of this evolved resistance. Due to the fear of this, several ideas have been proposed to A. slow down the growing antibacterial resistance, and B. make the antibiotics that we do have more effective. The aim of our experiment was focused within the latter objective. One common infection among us today is a urinary tract infection, where an individual's urethra is populated by bacteria that cause pain and urinary issues. When left untreated, the infection may spread further into the body, reaching the kidneys and the bloodstream, causing major and dangerous complications. One common bacteria causing these infections is Escherichia coli, a proteobacteria with a rod-like shape present on many surfaces and found in our gut flora. The easy access to this bacteria made it a prime specimen to conduct our research on. Our goal was to find whether or not temperature had a meaningful effect on antibacterial resistance in E. coli. To do this, bacteria cultures were grown alongside the antibiotic ampicillin in 3 different environmental temperature conditions of 37 degrees Celsius, 22 degrees Celsius, and 9 degrees Celsius. First, a culture was spread out on an agar petri dish. Then, a small disc soaked in our antibiotic was placed in the center of the plate. As the bacteria grew, there was a certain lack of growth in the center due to the antibiotic, defined as the exclusion zone. This was our measured variable, analyzed using the program Fiji ImageJ, where the area of the exclusion zone was taken as a percentage of the total area of the petri dish in which the bacteria were grown. The size of the exclusion zone represents the relative strength of the antibiotic in each temperature condition, showing at which temperature the E. coli are most susceptible to ampicillin. The results showed that the average exclusion zone size was larger by about 8% in E. coli grown at room temperature, 22 degrees Celsius, than those grown in ideal conditions, 37 degrees Celsius. The bacteria, however, exhibited no growth in the 9 degrees Celsius environment, as it was likely too cold, so these conditions were omitted from our graphing and tabulation. Using an unpaired t-test, our results were shown to be statistically significant with 9 degrees of freedom at a p-value less than 0.001, a strong correlation. With these results, one can make the conclusion that temperature does indeed have an effect on microbial antibiotic resistance. But what does this mean? The results beg for an additional couple questions to be answered. Why are antibiotics more effective in this way, and what can we do using this information? The full answer to both of these questions will take time to find, but by using this study and using the things we already know about antibiotics, we do have a good start. Antibiotics may work in multiple ways. They can prevent DNA replication by blocking important enzymes in the process, disallowing the bacteria from reproducing. They also may inhibit protein synthesis by preventing ribosomes from binding to the proper molecules to do so, killing the bacteria from the inside out. In this experiment, the antibiotic used was ampicillin, a derivative of penicillin, the first discovered antibiotic. Like penicillin, ampicillin works by attacking the cell wall, keeping the cell wall from completely forming. Temperature may have made this more effective by denaturing certain key proteins in cell wall formation, making it easier for the ampicillin to take its toll and eliminate the cell wall. Another possibility is that peptidoglycan, the main component of the cell wall, is easier to form in higher temperatures, bolstering the wall with strength that's not available at room temperature. Obviously, there are a multitude of hypotheses to be explored for finding the why behind the antibiotics-temperature relationship, so more research is required in this area. Even if we don't know the why just yet, we can think about how we may use this information once we better understand it. With the knowledge that antibiotics are stronger at certain temperatures, we may be able to administer them at different temperatures as well, enhancing their effectiveness in the midst of ever-strengthening bacteria. Future research may also provide further insight into how cell walls work, especially in their differences when grown in varying environments. This may allow us to devise new antibiotics that are more effective when applied at one specific temperature. For instance, one antibiotic may kill bacteria on room temperature surfaces extremely effectively, while another is more effective in body temperature conditions due to the different cellular wall construction. If research is continued and the antibiotic-temperature relationship is implemented within medicinal application, there are large implications that can be made. Realistically, bacteria will still over time develop resistance to antibiotics. This much is certain. The principles of natural selection are simply too strong to conquer, at least right now. However, using temperature as a tool may give us more time to study alternative methods of killing bacteria and ending the resistance crisis we have found ourselves in. I call on you, as a listener, to play your own part in slowing this crisis as well. Don't use antibiotics unless absolutely necessary, and always take all of the antibiotics prescribed by your doctor. It takes the individual efforts of the many to collectively slow antibiotic resistance so that the next generation is as disease-free and prosperous as humanity today. Thank you.